Good morning. On behalf of the organizing committee, Kathy Giacomini, Lawrence Lynn, George Scangos, Russ Altman, and myself, I'd like to welcome you to the inaugural Innovations in Regulatory Science Summit, which is hosted by the UCSF-Stanford Center of Excellence in Regulatory Science and Innovation, also known as Searcy. The primary objective of this summit is to convene a dialogue between stakeholders in innovation from academia, industry, regulatory agencies, the investment sector, policymakers, and anyone else who's interested in the advancement of innovation for medical products. We also want to highlight the power of Searcy and regulatory science, and you'll hear some lightning talks that will show some of the wonderful work that Searcy investigators are doing. And as with almost any other meeting that takes place this week in San Francisco, we hope that this meeting provides an opportunity for networking to stimulate collaboration between those who are attending. We'll start with an overview of the UCSF-Stanford Searcy from Kathy and Russ, who are the co-directors of the Searcy. We're fortunate today to have Stanford President Mark Tessier-Levine and UCSF Chancellor Sam Hawgood, who've taken time from their very busy schedules to make some opening remarks. We'll have keynote addresses from FDA leaders, lightning talks from Searcy investigators, panel discussions on accelerating trials, cross-sector collaboration, conflicts of interest, real-world evidence, AI, and bench-to-bedside research, to name a few topics. And we hope everybody will stay for the reception and poster session that will follow the program. We want to especially thank our sponsors, Novartis, Headland, and Beijing, and also encourage anyone else who's interested in sponsoring further summits or future summits to touch base with one of the organizing committee members. And a special thanks to our FDA colleagues who are here, some of whom will be on the faculty, others are in attendance. They made the very long trip, and fortunately there was no government shutdown this year, so we could have the summit. And I don't... And I also want to especially thank Rob Califf, who I think will be here later. Rob, when he was FDA commissioner, was very supportive of the concept of this meeting, and even as subsequently as a Searcy advisor, really played an important role in making this meeting happen. A few housekeeping items here. We have microphones in the front. There will be opportunities for the audience to ask questions, especially in the panel discussions. We have restrooms out and to the left. Wi-Fi is UCSF guest. And please silence your cell phones. Kathy and Russ are going to come up now and give us the Searcy overview.
Do I need to talk into that mic? What does this do? Which way does it go? So high. Okay, can you hear me? Okay, good. So first of all, I want to add my welcome to Cool Devs for all of you for coming, and I want to give a special thanks to our speakers who have managed to stick with us for a couple of years before we could host this event, so thank you all. Russ and I have put together a short PowerPoint presentation really to describe the activities of our Searcy with a focus on the research activities, although we'll be giving you a breadth of the other activities. And then secondly, to set the stage for today's meeting. And we're doing a dog and pony show, so I will talk and then he will talk, et cetera, then I come back. So I don't think I have to tell everyone or anyone in this room that FDA regulates a plethora, an enormous range of medical products, and those range from traditional small molecules, therapeutic antibodies, all the way to DNA diagnostic cell-based therapies, a myriad of medical devices, digital health products. And so for all this regulation, research is needed. And research in regulatory science is needed to inform the guidances, policies, and procedures that they need to have in order to review, evaluate, and regulate these products. And we're going to take a time, Russ and I decided we better define regulatory science because there's a lot of definitions floating around there. So we're taking a very simple definition for regulatory science, and that's research that helps regulators make better decisions across all the spectrum. So let's look at the life of an FDA scientist. So we know what traditionally we all think, what are they doing? Well, they're evaluating, they're regulating, they're monitoring, they're approving medical products, and that's what they do. But they have a very second important role that we feel are underappreciated, and that is they have a research mission. They have that mission to discover the knowledge that they need to inform the regulation of their products. And they often do this. In fact, if you go to the White Oaks campus, many FDA scientists are actively involved in research. In fact, some FDA scientists, that's all they're doing. They're doing research. Other FDA scientists have a dual role. They'll be doing research and they'll at the same time be doing review as well, and others simply do review. But in their research mission, in their active research programs, they often are doing it alone at FDA, but they also will collaborate. And they engage in collaborations with academia. And that has traditionally been a one-off thing. In other words, an FDA scientist will find an academic that's doing the research they're interested in, and they'll engage in a collaborative research project to help them inform their guidances and their procedures. But about a little over 5 years ago, FDA decided to fund a series or network of centers of excellence, academic centers of excellence in regulatory science and innovation, Searcy's, true translational research centers to work with them. So they put out an RFA, and a whole lot of people applied, and these four are the ones that are currently funded. So there's one at Yale Mayo, we've got University of Maryland, and we've got Hopkins, and then we've got UCSF-Stanford out here on the West Coast. And here in the audience, there are most or all of the Searcy's are in the audience. I've seen the PIs around here, and they will be presenting posters at the poster session, so please stop by. I think they've put some of their materials outside for you to look at what they're doing. We're going to focus our presentation on what's going on at UCSF-Stanford, what's going on at our Center of Excellence, and again, we're going to talk about research. And I'm going to turn it over to Russ to kind of give an idea of
Great. Yeah, thank you, Kathy. So this will be a romp through some projects. There will not be a lot of scientific detail, but it's to get you excited and see what the flavor and breadth of regulatory science is. We have had 42 collaborative research projects, which I would say all were FDA mission-driven. They're not really curiosity research, which is an important part of any portfolio. 28 are active, and all medical product centers at the FDA have been collaborators at one point or another. There's a great poster session at 4 where you will see many of these projects, and the posters are listed in your folder, I think on the left side. I said mission-driven, and that's an important thing because one of the successes of this, of course we want to write papers, but what we really want to do is impact regulatory decision-making, and one of the ways we do that is by impacting the FDA guidances and practices that are published and in some cases perhaps not published, but just the way science and regulatory decisions are made. And so impacting guidances is a real tangible and important goal in these efforts. So here are some of the areas of research that we've looked at: patient preferences, precision medicine. You can read these clinical trials. You can see that a lot of these are emerging, challenging areas for regulation because in many cases they're first-in-breed type products that are coming to the FDA. There is also a list of the research projects at our Searcy on the right side of your folder. So let me just go through a couple of these just to give you a flavor. How can the FDA incorporate patient preferences and experience into the evaluation of upper limb prosthetic devices? The project of Leslie Wilson, who you'll hear from later. But one thing to say about that is uptake of prosthetics that are very effective is limited by things like patient pain, things that are very reasonable reasons for a patient to reject an intervention. So let's look at what the real-world experience with these devices are and see if we can help fix that. How can the FDA ensure that drug labels are understandable, especially for pediatric drugs? For example, this is work of Lee Sanders and Bonnie Halpern Fisher. For example, they have told us that in some bilingual families, the parents may not be native English speakers, and so the label is primarily interpreted by a teenager or even a preteen, or even less aged, and there are challenges and because of that. How can the FDA improve the efficiency of data collection in clinical trials and develop novel statistical methods? So one of the insights here is that currently the record that is used for research and the record that is used for clinical care are often quite different. Imagine a world where they were the same record, and Laura Esserman and Steve Goodman have been thinking about this and other related questions in terms of evidence to understand how we can improve the healthcare system to actually help in regulatory decision making. How can the FDA use the electronic health record? This is on the same theme. And how can they improve pharmacovigilance? A tool my colleague at UCSF and our group has been doing some work in this area. We've been looking at FAERS reports, the FDA Adverse Event Reporting System, and have written AI algorithms that can prioritize the FAERS reports that look like they're high information content and highly impactful for early intervention and early examination by FDA reviewers, and then perhaps also evaluating the ones that look like they might be less useful. How can we develop methods to integrate digital health and monitoring devices? Auerbach, Lotz, and Roy are doing important work here, and they have found that even simple things like the lack of a universal identifier for medical devices that allow them to be tracked in the medical record is a huge barrier to collecting information about both efficacy and side effects of medical devices. Finally, work that's in Kathy's lab, you forgot to put your name on this slide, can excipients interfere with drug absorption, causing bioequivalence? So the excipient is the allegedly inert ingredient in your medications, but Kathy's group has done some work to find that some of these excipients are actually, in some cases, very potent inhibitors of uptake transporters or efflux transporters, and therefore blood levels can be markedly changed by some of these excipients. And they've created a database that's publicly available for companies and others to look at to understand this. So that's a quick run through of some of the research, and it really is meant to give you a feel for the range of centers and the range of questions that are being asked.
Thank you, Russ. Thank you. Okay, thank you, Russ. So in addition to research, because we have a Searcy, and I really think because we have the Searcy, we have advanced educational programs now at UCSF and Stanford. And we have a variety of different educational programs. I'm highlighting here some of our fellows, and there are a couple of them, I see them in the audience also, who have completed our fellowship program. And what's really unique about the fellowship program is our fellows rotate in academia, in industry, and at FDA, so they get a broad range of experience. And so if you want more information, I've put up the website there. We also have a robust visiting scientist program, and I have to say this has been a real landscape changer. So if you came to UCSF or Stanford, 5 years ago, you might occasionally see an FDA scientist here or there. Now, in the last 5 years, we've had 125 FDA scientists visit UCSF and Stanford, and they give public talks. So that's open to everyone, and we're telling you to sign up for an email on our email list if you need to know about that or would like to know about that. They also talk to our students, our faculty, and that's how some of these projects come about, is they have these discussions, and then the faculties engage, and then they start a collaboration. So that's also been great. So how can you participate? So our purpose in today's, as CoolDev said, our purpose in today's symposium or summit is really to engage industry now and engage the private sector in what we feel is the most exciting thing happening here between our campuses and FDA. So enroll in our courses, volunteer to teach, watch our Searcy channel, attend our seminars, attend future conferences, and participate in research. I'll tell you about how to do that. First thing you have to do is sign up for our email list if you're not on it. So go to our UCSF-Stanford Searcy site and sign up. And then today we are launching a call for industry participation in Searcy research projects. So those projects, and you'll hear more from Janet, I believe, later when she gives her keynote, those projects are solely funded by the limited dollars that FDA receives for research. And then they're done in collaboration with us. We would now like to have the third group of people who are quite interested in regulatory science industry involved. So we've put a little form in your in your packet. And
Right side.
The right side, thank you, Russ. And we're asking you to complete it because we want to now involve industry in these research projects in a collaborative way. So. And then I'm not going to repeat what CoolDev said. We have a really exciting day with several speakers, all sorts of lightning talks, two keynote speakers, both Janet and Peter Marks, and we're very excited that both of them are here. The heart and soul will be the panel discussions, and I will emphasize that what we're really striving in those panel discussions is the panelists will give remarks, and we would like a lot of participation from the audience and a lot of Q&A. And those panel discussions are really designed to think deeply about science that is needed to regulate drug products. And then I want to end by thanking everybody, the organizers, CoolDev, George, Russ, and then, of course, Maria Fricello and Lawrence Lynn, wherever he is. There he is. Can we give him a hand? He has really done the heavy lifting. And then I'm going to say it once, but we'll probably say it again and again. We've included really nice bios in your packet, so we're not going to have long introductions to all these stellar people who are speaking today. So next up are the president of Stanford University, Mark Tessier-Levine, and Sam Hawgood, and they don't need any introduction, and if you'd like to, you can read about them anyway. So please come up. Thank you.
Great. Well, thank you so much, Kathy, for that introduction. I want to thank you, CoolDev, Russ, George Scangos, and all the other organizers for bringing us here together for this important meeting. We're here today at a time of both great medical need, but also great scientific opportunity, a time when today's discoveries become tomorrow's medical advances. These advances hold the promise of transforming care for difficult, stubborn conditions that have afflicted patients and frustrated researchers for decades. Now, together, the academic, industry, and regulatory sectors are the engines of this innovation, and the enhanced effective collaboration that this summit promotes can boost performance on behalf of patients everywhere. I'm actually really excited that this summit brings together all three sectors to the table, and the issues that will be discussed will speed access to crucial, safe, and effective medical products. The discussions will also help us understand more about the specialized, often unique challenges and complexities involved in regulatory science and medical product development. We're going to be digging deep today. In my comments, however, rather than digging deep, I'd like to really step back a little bit and provide a broader frame and touch on the importance of the alliance among these different groups, why they work, why no one sector can do it alone, and what makes the Bay Area a special place for this sort of collaboration. Now, at a time when there is so much bad news out in the world, it's important to remind ourselves that America's last century of research, education, and scientific exchange has been a glorious story of leadership and innovation. That leadership has been built on a powerful tripartite partnership forged among academic researchers, innovative companies, and the federal government. Scientists in our universities and research institutes churn out discoveries, much of them supported by government funding. Then companies build on those discoveries. They develop therapies and cures for serious diseases, and they work hand in glove with government regulators to bring them to patients. History shows that this sort of collaboration can help sharply reduce or eliminate major causes of morbidity and mortality. Think of the decrease in mortality from heart disease and stroke by over 50% in the past 40 years. HIV/AIDS has been transformed into a disease that's manageable without hospitalization. And although death rates from cancer still remain too high, we have been seeing encouraging progress in recent years, including, as announced just a few weeks ago, the sharpest year-over-year decrease in mortality rates, which is a great evidence of progress that is being made. The human benefits, of course, are enormous. So are the economic benefits, estimated to be in the trillions of dollars from these advances. But we all know that huge health challenges remain. We're still only partway down the path to conquering cancer. Think of the infectious diseases that are becoming resistant to our arsenal of antibiotics. Think of chronic diseases like Alzheimer's that are on the rise in the case of Alzheimer's because of the aging of the population, and the suffering of the patients, the toll on their families and loved ones, and the economic toll through cost of care that could bankrupt us. There are a host of other challenges facing us, and we can't delay. But no single group can bring a product to market alone. Academia, industry, and government must work collaboratively and make it easier for each sector to do its job in a timely manner and avoid excessive and unnecessary hurdles. Strategic, effective collaboration enables innovators in academia and industry to know what's expected when new drugs and therapies are reviewed for safety and efficacy. That leads to my second point. Innovation can lead to discoveries, but those discoveries are meaningless unless they reach the patients who need them, and especially if they reach the patients in a timely manner. We all know that it can take well over a decade and several $billion to go from an existing scientific insight to a drug on the market. And even after all that time and expense, not all therapies work well. Everyone has experienced the twofold frustration that comes when a promising trial fails, but not before it's vacuumed up resources that might have been used to help develop another therapy. Greater communication, greater interactions among academia and regulatory bodies, such as the FDA, can help companies develop better and more cost-effective methodologies. So this leads me to Searcy also, and we had a very brief outline of some of the exciting work done at Searcy. One of Searcy's major goals, of course, is to identify less onerous and less costly ways, but sufficiently rigorous ways to meet the standards the FDA is charged with upholding. And the FDA can use that knowledge of the challenges and costs in weighing whether and how to reduce barriers. Although the process involves many complexities that all of you here are involved in on a day-to-day basis, the bottom line is relatively simple. If we can test a drug and see if it works more efficiently without sacrificing quality in the review process, this can lead to less expensive drugs and can make them be brought to market much more rapidly. Can reduce costs, free up funds to pursue even more treatments, and researchers can take on even more challenges. And so what I'm excited about is the type of work that Searcy is doing to explore ways of accelerating and improving the whole drug discovery process. We saw some examples today. Others include looking at the use of smartphones to submit real-world evidence from patients' homes, focusing on how to gain broad agreement on adaptive clinical trial design. The use of AI was mentioned by Russ. It can be used in a whole host of different settings, including to improve clinical trials and use the wide net of big data to help identify and enroll subjects for trials more effectively. Now, if we're to explore these possibilities adequately, collaboration is a must-have, but opportunities for such conversations are scattered and too few. And so my final point is to just celebrate the special opportunities here in the Bay Area for collaboration. Collaboration has been a way of life for us at Stanford throughout our history, and we're proud to work with our UCSF colleagues through Searcy. From my own time on the faculty at UCSF back in the 1990s, and also from my interactions with Sam Hawgood, who you'll hear from in a few minutes, I know and deeply appreciate our shared commitment to regulatory science. I think that together we're specially positioned to serve as a liaison and to provide support, not least of which includes discussions today and our extensive interactions with the FDA. As well, our universities, of course, are situated among a host of vibrant pharmaceutical, biotechnology, and high-tech industries, an array of schools, departments, and institutes with a vast capacity for ideas and support. The interactions between our universities and the FDA have brought many FDA scientists here through the FDA Visiting Science Program, brought more than 124 FDA scientists to both our universities. They meet with faculty, give public talks, they visit local companies and organizations, and in turn, Searcy provides speakers for a monthly lecture series for FDA staff. Searcy and FDA have organized workshops that spotlight natural language processing, patient preference, clinical trials for breast cancer, and many other topics. This type of collaboration really supercharges the skills of researchers. It amplifies the impact of the solutions they achieve. And together, through research, innovation, and collaboration, we can help improve life for millions of people in our country and around the globe. Now, the world has come to rely on the U.S. taking the lead. We can set the pace and set the right example with top researchers in academia and the research world, with vigilance and proper safeguards by government regulators, and with companies who can run with discoveries and get them to the patients who need them. Together, we can look forward to continued vibrant research and the life-saving benefits that will flow to our communities, our nation, and the world. So thank you to all of you who are here today. Thank you to Searcy and its leadership and our colleagues from the FDA who are here today as well for the roles that you play and your commitment to taking what's good and making it even better. Thank you very much.
Well, good morning, everyone. It's a pleasure for me to welcome you to our Mission Bay campus here at UCSF and to follow my friend and colleague Mark in remarking how important the work that Searcy is doing and all of you who are here today expressing your interest in that work. It's fitting, I think, that the meeting is being held in the Rudder Center, named after Bill Rudder, a faculty member at UCSF for many years, who some 40 years ago pioneered molecular biology towards vaccine development and made huge changes in the world's health with the hepatitis B and hepatitis C vaccine. And 40 years on, we're now pushing for further advances in biology. I'd like to begin by thanking Kathy and Russ for your tremendous leadership in Searcy over the last several years. I know last year we were going to have this meeting and it backfired for reasons that were totally out of your control, but thank you for hanging in there and pulling together what looks to be a wonderful, wonderful meeting today. I would also like to thank the leaders from the FDA who are here for their vision some 5 or so years ago in conceiving of the concept of research in regulatory science and putting the Searcy programs together and continuing to fund and support them, and particularly those leaders who have taken time out of their busy schedules to be here today. So thank you to the FDA. I'm going to echo briefly, because I think Mark laid out beautifully the reasons that you're all in this room today and the critical importance of the partnerships between academia, industry, and the government. So I'm not going to repeat everything he said, but I would like to pick up on 2 themes that he discussed. And for me, when I think of where Searcy sits in the UCSF landscape, 2 words are important to me: time and place. Time, because I think we're in an incredible point of acceleration in science at the present time. Work is being done that will translate into better cures for patients. I like to think about progress in science using a paradigm that Stephen Jay Gould put together for evolution, and that is that although we build on the shoulders of those who came before us, progress in science tends not to be linear. There are particular points in history where he described what the term he used was a punctuation in the equilibrium of evolution. And I believe we're in one of those punctuations in science right now where progress is accelerating at an exponential rate. So there couldn't be a more important time for regulatory science to be operating. Place, as Mark pointed out, the Bay Area is known throughout the world as an ecosystem of innovation. Great universities, UC Berkeley, UCSF, Stanford, UC Santa Cruz, 2 national labs, Lawrence Berkeley National Lab, Lawrence Livermore National Lab, and of course, just wonderfully innovative companies, both in pharma, in biotech, and in tech. So bringing time and place together, I think it's critically important that this partnership between Stanford and UCSF exists to make sure that the regulatory science can keep up with the pace of discovery in overall science. The last point I would like to make, and I hope it's thought about during the day as the panel discussions are taking place and as we think about bringing science to patients, is the issue of health equity. There is no question that there are enormous advances, whether it's finally being able to understand how we can take the human genome and think about that in a translational clinical delivery model. We can now write the genome. We're now talking about a whole new generation of living therapeutics, turning cells into effectively engineered robots to survey and deliver their actions. But we must think about all these wonderful advances in the light of health equity. Here in the Bay Area, one of the richest areas in the world, we still see pervasive, refractory issues of health equity, whether it's in the prevalence or the outcomes of cancer, neurologic disease, diabetes, obesity. These things exist right in our backyard. And I think it's critically important that we keep an intense look on that as we think about how to move these wonderful discoveries into the clinical arena to make sure that they're both accessible, affordable, and deliverable to everyone in our population, not just a few. So with those remarks, again, I welcome you all here. I drove into the car park this morning at 7:30 and I thought, wow, I'm going to have to go up to the top floor here to get a park, and it just reflects, I think, the commitment that you all have to the work that you're doing, and I know it's an incredibly exciting program, so it's a great pleasure for me to welcome you to UCSF and enjoy the rest of the day. Thank you.
Okay, so we're going to get into the program, the scientific content. It's really very simple. They're either lightning talks or panels. And our first one is, and we tried to have them thematically related. The first lightning talk, entitled Defining the Strength of Evidence in Therapeutic Development, will be given by Stephen Goodman from Stanford.
Great. Thank you very much. So I see we're ahead of time a little bit, so maybe the lightning part of this doesn't have to be quite as rapid. I have to say, it's very daunting to give a lightning talk in this context because, what do you think of when you think of lightning? You think shocking, electric, blazing speed. If you ask someone to describe the FDA, those wouldn't exactly be the first words. But we will, that will be one of the themes here is how we can push them closer to honoring those principles, and I'll try to do my best. So this is a project where we were challenged, proposed, and then accepted to help define or redefine the strength of evidence metric that's used in therapeutic development. And this is work jointly with myself and my postdoc, Perrine Geniot, who's here in the audience. She'll be in front of the poster later, and she's done some yeoman's work, as you will see. So I just want to start with the regulatory, the historical context, and I'm not going to spend much time here, except to note, of course, in 1962, we started on this journey with the Kefauver-Harris Amendment that added efficacy to the statutory mandate for the FDA, and in that was a requirement for substantial evidence of effectiveness. that is evidence consisting of adequate and well-controlled investigations, which later was interpreted through guidances by the FDA in a way that we will talk about. Now, I will say that there are several features of this rule. Oh, let's see. Okay, there was a slide here that, let's see if the next one comes up. Okay. That it aimed to minimize the probability of false positives. So the way this was interpreted was as two positive trials. And this was very, very important. The next slide was actually of my clothing, because what I'm wearing is something I affectionately call my FDA tie. And it's a picture of the nostrums and elixirs that used to be sold that were worthless and often dangerous before this amendment came in, and that are now effectively off the market. So the cornerstone of this rule of efficacy was two positive trials. And the aim was to minimize the probability of false positives due to bias or misleading evidence. And one of the advantages is that it was very operationally clear. And that has served the agency very well for a long time. But even as late as 1998, in the guidance on clinical evidence, we see that what happens when one wants to go lower than this threshold, it's described in this way. It says, a single favorable study among several similar attempts that failed to support a finding of effectiveness would not constitute persuasive support for a product unless there was strong argument for discounting the outcomes in the studies that failed to show effectiveness. Now, I'm going to show you what's wrong with that logic. It seems on its face to be sort of reasonable, but here's the other way to look at the two positive trial rule, which is it's actually a form of meta-analysis. That is, it's a method of evidence synthesis, which is vote counting. But the problem is that it's a form of meta-analysis that's statistically flawed. And on the left, you see an array of studies, 4 studies, 3 of which are not significant, 1 is significant, and you see them added up on the left. They count for 0, 1, 0, 0. So the evidence score through the two positive trial rule would add up to 1, even though these all aligned would come up with an effect measure that is actually highly significant. On the right, we have a 2-win, 2 trials that are statistically significant. Let's see if I can get my little arrows here. Let's see, you don't see my cursor. Okay. Anyway, and you see on the right, the evidence score is 1, 1, and then the 2 that are not significant are 0, 0. So this adds up to 2. So by vote counting, the evidence in situation B is much stronger than in 1, because we have 2 wins, but using just very standard conventional statistical techniques, the evidence on the left is much stronger. So in fact, the 3 trials that are not significant don't have to be discounted. In fact, they have to be included and properly added up. In case you think this doesn't really happen in real life, here's one of the most famous meta-analyses that were ever done, Richard Peto's meta-analysis of the use of tamoxifen, adjuvant tamoxifen for breast cancer. And you might not have appreciated that in women over 50, this shows the evidence base, 23 clinical trials, only 1 of them was statistically significant. So it has an evidence score of 1. But in fact, as you may know, this showed that there was a 20% mortality reduction with a p of less than 10, 1 in a billion. So this is a pretty definitive study, and yet there's only 1 win among the 23. So features of the two positive trial rule is that it's an imperfect evidence summary leading to ad hoc adjustments without principled justification. It's hard to establish clear or consistent FDA case law on the basis of this. And it incentivizes, because the trials are all or nothing, it incentivizes all the things that FDA mightily struggles against. Outcome switching, all sorts of post hoc chicanery in a trial that was negative, evidence suppression, because they don't want to, they don't have to show all the evidence, and gaming. It's also remarkably inefficient because it involves throwing out some of the highest quality evidence that you have. That is the evidence from RCT, phase 3 RCTs, and they are not incorporated into the score. Now, we have the benefit of FDA's latest thinking on this, and I will say that it's easy to poke at the rule, but in fact, in practice, FDA has been very, very flexible. And looking back over the cases over the last decade, they don't adhere strictly to this rule at all. And I have to say they have to be lauded for that. There's a lot of flexibility. And this guidance, which just came out several weeks ago, it's a draft guidance, sort of summarizes their current thinking on the topic. And it's a draft guidance, so we'll see how long when it's finalized and what will come out of that. But it's still very, very useful. And I want to say that I look at it as very, very positive. So there are a whole list of considerations that could be considered in deviating from the 2-trial requirement. That is, if you have a large multi-center trial, and this was also included in 1998. Some of these are carrying forward from 1998. If you have evidence from related indications, if there's strong mechanistic support, if there's an established natural history, if there's evidence from similar drug classes, serious outcomes with an unmet medical need, if there are rare diseases where you just can't get clinical trials of adequate size or at all, disease subsets. Every disease these days is a rare disease. And finally, if the efficacy trial is unethical. So all of these are to be applauded, and they're already being used by the FDA in numerous decisions. I want to point out all the judgments that this guidance calls for. And again, I'm not saying that these are bad, but these are all, in some sense, undefined terms. There's very little quantitative in this guidance. Persuasive statistical effect, which renders the second trial unethical. A strongly positive trial. A robust statistical effect. Inconsistent statistical evidence. Sometimes that's interpreted as significant in one study, not significant in another study. Compelling efficacy results. Strength of evidence, but no definition. Efficacy that could be fairly and responsibly concluded by experts. Interesting. And in this last one, and again, I actually think they're struggling with very real issues here. This is not in any way to denigrate what they are struggling with. I'm going to offer some proposal to maybe help with that struggle. And finally, in all cases, this is a quote, in all cases, FDA must conclude that there is substantial evidence. That is a statutory standard. So we cannot relinquish the phrase substantial evidence. However, the degree of certainty supporting such a conclusion may differ depending on the clinical circumstances. So now a line is being drawn between the strength of evidence and the degree of certainty. Very difficult. I will say that there's no definition nor quantitative measure of this certainty. The only mention of statistics comes under the one paragraph on statistical considerations. The first sentence is the classic 2-trial rule, with significance being defined as p less than 0.05. It says, a lower p-value, for example, would often be expected for reliance on a single trial. And then, for a serious disease with no available therapy or a rare disease, a somewhat higher p-value, if pre-specified and appropriately justified, might be acceptable. I would challenge anybody in this rule to figure out exactly how they could justify and exactly how they would adjust it up. We do it in a fairly ad hoc manner. So the question is, can a formal evidence score help? We already know the qualitative characteristics that we want in a more rigorous and formal approach to this process. So evidential strength is modified by study design and drug features. So we already know from this guidance and just from our own knowledge about scientific methodology that a large multi-center RCT is a strong design. So that's a positive. If it's observational study, that's negative in terms of strength of evidence. If it's based on surrogate endpoints, that weakens the strength of evidence. If it's short-term outcomes, that weakens the strength of evidence. If it's a validated mechanism, that strengthens the weight of evidence. And also the degree of certainty that's needed is contingent on the seriousness of the disease and the extant therapies. So an orphan disease without an alternative, we might want to set a lower evidential bar. A Me Too drug with multiple alternatives, a higher bar. A breakthrough drug with better safety, a lower bar. A serious condition, a lower bar. And those with a safety concern, a higher bar. So we know the qualitative dimensions that we want to incorporate. And again, these are reflected in FDA conversations. So we set out to create an evidence score based on a sort of Bayesian logic, which provides a valid calculus of evidence. So Bayesian methods, and I'm not going to define them here, are often thought to be some sort of calculus of belief or certainty. That what they actually are, the way that I look at them, is a calculus of evidence, and they're a proper metric for evidence. An evidence score can incorporate all the factors that were just listed to justify flexibility, and it's on a quantitative scale that's like a Rosetta Stone. That is, it can be translated into degrees of certainty, into strength of evidence in terms of p-values, prediction value, probabilities, or Bayes factors. Whichever language you speak, this score can be transformed into that. This sort of display shows how difficult it is to take the conventional metrics and translate it into certainty. So this is just really, really simple, basic Bayesian logic. So the certainty of a non-null effect, if you have an RCT, a perfectly done study with a p of 0.05, if you start with the probability of that drug being effective being, I'm going to go in the second line down, of only 10%, and usually those are not brought to the FDA, we'll say, a p of 0.05 at most can bring you up to 43% certainty. This is just to show you that certainty is very different than the strength of evidence and the number embedded in the p-value. If you're bringing an unlikely drug or with an unlikely or unknown mechanism in a disease that's been notoriously resistant to successful therapies, which might have a 25% chance of being effective, a single p of 0.05 only brings you up from somewhere from maybe 45%, that is less than 50-50, to 69%. And finally, if the drug is a 50-50, that degree of certain evidence brings you up to something like 70% to 87% chance of the therapy working. This is the actual probability of error that we should be worried about. But we don't have a language for it. So what did we do to establish, both come up with the factors and establish an empirical base? We looked at all of the applications that had been considered between 2013 and 2018 that had received at least one a complete response letter. That is, that they were going through multiple cycles of review. And the reason is, in order to develop the metric, we have to have cases where the FDA says we're not going to approve it, and cases that we are. So we could show where the score, where it was insufficient and where it was sufficient. Unfortunately, as outside contractors, we have no access to information on drugs that are disapproved. So we don't have access to that evidence base, but we do through the drugs that have gone through multiple cycles. And that information does exist on FDA.gov. And this is the yeoman's work that Perrine did. She poured through the briefing documents, the advisory committee minutes, the transcripts, the approval letter, the summary review, the office director memo for all of the agents that went through multiple cycles of review looking solely at efficacy, so where efficacy was the only issue. So we had 22 of those cases out of the 912 total. Wasn't important the number, but that these provided sort of the test case for developing a score. So I'll just show you how it would work with pirfenidone for idiopathic pulmonary fibrosis. I'm not going to show you all the details here. We haven't finalized all the weights and components of the score, so I didn't want to show you all those results. I'll just give you a hint. But this was the first cycle. The primary endpoint was mean change in percent predicted forced vital capacity at 72 weeks. So actually that's pretty good in terms of time point. And unfortunately you don't see the submitted studies very well, but there's two studies. One is statistically significant, one is not. So in cycle one, there was only one study that was that sort of counted, but it's a life-threatening disease, and mortality was actually in some ways of more interest But it was a secondary outcome, and it failed the statistical criteria, which were a p of 0.01 for that. And the components of the evidence score that put together was, A, was this randomized? 2, was the endpoint measured sufficiently far out? And that was positive on both. Did they use a surrogate endpoint? And there's a little cancel sign there, but it just means that that counted negatively. Is it a life-threatening disease? Yes, or orphan designation. And finally, is the mechanism of action understood? And in this case, it's not really, not to the extent that it would provide actual evidential weight. So the final evidence score for this cycle was 5.9. 1.9 was just the pure statistical component, is literally a translation of the p-value from the meta-analysis of the two trials. Everything else came from the other more qualitative aspects. So this resulted in the complete response letter. Cycle 2, an additional trial. That's the panel below. It's very faint, I'm sorry. The additional trial showed actually pretty dramatic effect on forced vital capacity. And this dramatically increased the statistical component of the evidence score. Everything else stayed the same. So the final evidence score was 18.9. Now I know these numbers will not mean anything to you. I'll start to go a little in translating them, but as we change the weights, going forward, this will change. The important thing is not necessarily the score itself. Of course, temperature doesn't mean anything. If I tell you the temperature out is 23, that doesn't tell you anything until I tell you, is that centigrade, is that Fahrenheit? And even then, it doesn't have meaning until you experience it. You have to use it. All metrics have meaning through use. So if we start using these things, they will start to have more meaning. So that was approved. So this is just an example of the trajectory of the evidence score as we currently have it configured for 5 different agents. You will see that all of them start at a low level and all of them increase, and all of these were approved in the second stage. And each one of them has a complex story, and if you're really quick on your feet, you will say, the one at the top, why wasn't that approved right off the bat? Because it seems to start in the area where the other ones finished. So the good thing here is everything is, towards approval is going up. But the issue for the top drug, which was tolvaptan, was the endpoint used at the beginning, which was, this was for kidney disease, autosomal dominant cystic kidney disease, was decrease in kidney volume, and that was deemed not actually sufficiently clinically relevant. So they wanted to change the endpoint. So this is a strong score, but for an endpoint that was not deemed optimal. So the endpoints were changed, a new trial came in, and in fact, as you can see from the memo, that they really lauded the design of the second trial. For the one at the very bottom, which is droxidopa, you see barely a change. They actually changed the endpoints here as well. So we're not perfectly capturing this, but the fact that it was a very low score, and I would put on this metric the approval threshold somewhere in the range of 10 to 15, and I actually don't want to substitute the p of 0.05 for a bright line here. That will induce the same sort of gaming, but this is at least an objective way to represent the evidence. This was seen as very, very difficult in the agency. It says there's no doubt that the data are, quote, at the margin for approvability. So this was seen, and it's reflected in the score, as very, very close call. And I think the orphan status and the seriousness of the disease is what pushed it over the top. So in conclusion, the evidence score starts with a formal meta-analysis of the extant evidence, which is a valid method of evidence combination. It may require a more complete submission package, though, if the negative studies count, which they should. And in fact, if we're talking about efficiency here, which we're going to talk about all day, and the inclusion of all this indirect data, mechanism, similar drugs, et cetera, we cannot afford to throw out the highest quality data that we have, even if it is statistically not significant. So this should be the first target of our attentions, is how to use all of the data from phase 3 trials and not negate it. The qualitative factors in the evidence score are all already used, but informally. So it's very difficult from case to case to say exactly how much each weighed, exactly how do you bend the rules, and by how much. So this, as I said, makes case law really difficult. Everything is bespoke. And it would be nice if we could develop a backbone where you still have some bespokeness, but some of the elements of the contributions of the decisions are clear. So the evidence score does not replace judgment, but it does or can improve rigor, transparency, and consistency, I would argue, and efficiency of FDA efficacy assessments. So that's it for the lightning strike. I want to thank my FDA collaborators who we have been meeting with regularly. They've been enormously helpful. Estelle Russick-Cohen, Talbot Irony, Tom Permut, and Lisa Lavange are no longer at the agency, but they were incredibly helpful, Lisa in particular, in getting this started. Janet Woodcock in supporting this strongly, both financially and morally. The Searcy itself and the Meta Research Innovation Center at Stanford, of which Perrine is a fellow. So thank you very much.
Thank you, Steve. I think we're all thunderstruck. That's a pun. So Steve will be on the next panel, so I'm going to allow us to go right into the panel session, and you can ask questions during the Q&A. The panel sessions will have the structure, and I'd like to ask the panelists to come up, please, and take their place. The panelist structures will have introductions by our moderators, who in this case are Laura Esserman and Janet Woodcock from the FDA, and Laura's from UCSF, and then a few comments from the panelists, and then we guarantee a robust Q&A at the microphones. I just mentioned our moderators. The panelists are Amy Abernethy from the FDA, Steve Goodman, who you just heard from, Sandra Horning, former Chief Medical Officer at Genentech Roche, Carl Peck, Chairman of NDA Partners, and Alfred Sandrock from Biogen. So I will throw it to our moderators. Thank you.
Great. Thanks, Russ. Well, good morning, everyone. It's great to see such a big crowd on an early Sunday morning. I am Laura Esserman. I'm a professor of surgery and radiology at UCSF, and I'm the director of our multidisciplinary breast cancer program. My interest in innovation and trial design comes from running an adaptive platform, randomized platform trial called iSpy, where we're using neoadjuvant therapy in breast cancer for early-stage high-risk patients as an example to show how we can accelerate the pace of getting the right drugs to the right patients at the right time. And as well, I run a large national study called the Wisdom Study, which is looking at mammography screening and comparing a personalized approach versus kind of a one-size-fits-all approach to see whether we can drive innovation. And in these contexts, I think a lot about how to innovate in regulatory science and to try and figure out how we can make our science more patient-centered and how we can develop approaches that actually work for industry's devices, advancing science, but really are taken from the perspective of how to improve both an individual patient's outcome as well as the outcome of populations. Over the years, I've had the great fortune to work with Janet Woodcock, who's been a real inspiration to me, and I think someone who is a true big systems thinker, and I think is a national treasure at the FDA, and I hope she stays there forever. So with that, I'll turn it over to Janet.
Well, I'm glad this panel is on the clinical trials. I believe the clinical trial system is broken and we have to reform it. And so I'd be quite interested we have a bunch of experts here. I'm the director of the Center for Drug Evaluation and Research at the FDA, and I've been at the FDA a long time and been observing what the clinical trial system can and can't deliver, and we really need to do better for our patients, and we can do better. Those techniques are available. I would like to say to Steve, thank you. I think you're on to something. I believe this trial counting, onesies and twosies, this has driven me crazy for longer than I can remember. I do think, though, that your surrogate endpoint, you're going to have to put something, your examples even showed that. You're going to put something about the quality of the surrogate in there as a metric.
I object to that.
Well, actually, we have that in there. We have the validated surrogates and the non-validated surrogates.
Yeah, but, there's more to it than that.
Well, that's where judgment can come in.
Oh, okay. Okay. So I really look forward to this panel. Laura's going to tell you how we're going to set this up. We have lightning comments, and then we're going to leave a lot of time for discussion.
Exactly. So we thought to make this interesting and innovative, we're going to give each panelist 3 minutes. That includes a brief introduction and the answer to question 1. And then we're going to go on to we'll do a lightning round of question 2. And then we're going to try and see what comments resonate the most. And Janet and I also get to answer the questions. But so question 1 is, what is your top suggestion for an innovation that could accelerate drug development and thereby increase the public's access to effective and cost-effective treatments? And if you haven't stopped talking by the time the alarm goes off, we will move on to the next person when it does. Oh, wow. Amy.
Oh, here we go. Amy Abernathy, Principal Deputy Commissioner and Acting CIO at FDA. Prior to this, I was in the health tech industry, and prior to that, I was a professor of medicine and oncologist at Duke University. With respect to what do I think is going to be a key innovation, we've already heard about work being done in the Searcy's that, for example, look at the use of electronic health records to identify adverse events. We look at the use of smartphones for patient-reported outcomes, et cetera. We can do innovative development of what is possible, but ultimately we don't have in place currently a trust infrastructure for how those innovative developments then ultimately get to scale to be used across. What do I mean by that? Ultimately, we need companies that are building products that are then bought by, for example, the life science industry and others to then be used across the clinical development and clinical research enterprise. And those companies need to know, how can I be confident that this is being built in a way that's trustworthy to the overall clinical enterprise, including the FDA? Because if it's not trustworthy within that enterprise, then it's not going to be purchased, and it's really hard to build those products. So I think that one of the key innovations we need is a trust infrastructure. As FDA, we're starting to try and think about how to get there, but we need this entire community, including the Searcy, to help us understand. Practically speaking, we need to understand what's the distribution of products that need to be developed and where that needs to go. For example, solutions for data curation for real-world evidence, distributed clinical trials, monitoring of drug manufacturing, even in the food space. And then we need to understand, what does good look like within the context of each of those kind of products, and how can product innovators build products in line with good. So that's where I would put my innovation. How'd I do?
Congratulations. Excellent. All right. Steve.
Well, I almost feel like ceding my time here because I've already had more than my share. I'll just say 2 quick things. I think it'll be well under 3 minutes. First of all, obviously, the biggest bang is not in the reducing the time for the approval process, but reducing the time for the drug development process, which is far, far longer than the approval process. Anything that will use all of the information extant and not any clinical trial designs which can experiment, focus their resources most effectively on those who are most likely to benefit, and also minimize randomization to groups for which we already have a lot of information, which actually iSpy is one of the models, which borrows as much information as possible, is going to increase the efficiency of the operation. So anything that involves information loss in the system, that is experimenting on groups or on comparison groups, that is not giving us much information, is slowing down the process. So I think better use of adaptive designs and better funneling of the most highly promising therapies into the final confirmatory pipeline would get us far. The second part of that, yes, is the approval process. And I think that often FDA is very risk-averse because of the irreversibility of some of its decisions. Some of the expedited pathways do have required confirmation, particularly for surrogate endpoints. But I wonder if this model that is akin to evidence under coverage under evidence development, as some of the payers have used, which you can have an expanded scope of provisional approvals based on continuing evidence, might change the calculus at the FDA. I think the FDA doesn't want to make a mistake, and I completely understand that, but if the stakes for those mistakes in certain situations could be lowered by having this sort of provisional approval, which would then be enforced, the big Achilles heel is requirements that are then never fulfilled. And maybe this is already being discussed in the agency. This might allow them to make quicker decisions in a provisional way, which then in some cases would be pulled back, hopefully not very often. So I'll leave it there.
All right. Good job. All right. Sandra, you are up next.
Okay. Well, as introduced by Laura, I'm the former Chief Medical Officer and Head of Global Product Development in Genentech, following a lengthy academic career at Stanford. During that 10-year period of time, we had 15 new medicines approved, over 30 line extensions, and 28 breakthrough designations. And on the basis of that experience, I think I would agree with what Steve said, which is ways in which the development process can be streamlined. And they are numerous, but I tried to pick one for the sake of discussion and to be provocative. And that would be to really establish meaningful clinical endpoints that are intermediate or surrogate. And working the hard work, doing the hard work as a community in developing and validating these. If they were done and if they were available, they have many utilities. Those would include making faster and better decisions throughout the development period of time. We'd have a better handle on how to size and design our trials. We could go into adaptive designs with a lot more confidence and potentially even as an industry be willing to cede some of those decisions to independent bodies. And I think as we make more and more progress in very difficult fields like cancer, we're now beginning to see, as Mark just stated and we've seen in the news, real meaningful differences and advances in cancer. Now we have to move much earlier in the disease. And in order to do that, I think we're going to need these intermediate endpoints. We've already made some progress in pathologic complete response, thanks to Laura and Janet. It's taken us 10 years for minimal residual disease in hematologic malignancies, and I think we're still not there. And we're really on the verge of circulating tumor DNA, which I think can change the calculus for solid tumors. So with that, how'd I do? We'll stop and go next.
Yeah, actually.
Okay.
Great. Less than 2 minutes.
Oh, all right.
Awesome. All right. Carl, are you next? All right.
So I'm Carl Peck. I am Janet Woodcock's predecessor in this job. I must say I admire her staying power and endurance, but most especially her support for innovation in drug development and regulatory science. I'm going to take a slightly different tack and actually answer the question with respect to access and cost. The focus I anticipated would be primarily on innovative new drugs that would be breakthrough, beneficial to unmet medical needs. But the fact is 90% of prescriptions in this country are filled by generic drugs with very significant reductions in cost to the patients. And yet there are still hundreds of drugs that are off patent, off exclusivity, that are still costing too much because they are not generic, and there are significant barriers to the development and approval of these generics based upon the standard generic drug approval paradigm, which is a simple pharmacokinetic test. These include, if I can pull my notes up, these drugs that are waiting to be genericized are orally inhaled and nasal products. such as Advir, ophthalmic products like cyclosporin, ophthalmic solutions, some GI products, complex dosage forms, long half-life injectables, numerous cancer drugs that have long half-lives that are not amenable to the quick bioequivalence test. Complex drug-device combinations, such as meter-dose inhalers. It's notable that it took 10 years for the EpiPen generic to meet regulatory standards. And complex active ingredients, peptides, polymeric compounds, and so forth. Fortunately, the Congress has recognized this, and FDA has recognized this, and there have been 2 generic drug prescription fee authorizations that have enabled Janet to support advanced research within the Center for Drugs in the generic drug division. And my time is down to 20 seconds, so I'm not going to be able to tell you just how extraordinary the research activities are in that group, but they are applying sparse sampling, population PK/PD, modeling and simulation, Bayesian estimations, AI, machine learning, and so forth, all applied to advancing the capacity to do the trials or the evaluations that are necessary to. So my top suggestion is to intensify and support research and application of advanced trial and analytics for generic drugs.
Okay. Great. All right.
Thanks. My name is Al Sandrock, and George, thank you for inviting me to speak here. So what I'm going to talk about is really not an innovation, but it's using a technology we already have better. So what I'm proposing is a new academic discipline called either quantitative medicine or clinical measurement science, and also separating treatment from measurement assessment in patients. Perhaps next door to the hospital, you would have what would be called a measurement center. So let me take you through, and I come at it from the company point of view, where we waste, I think, a lot of time and money on clinical trials. Most R&D heads will tell you that the budget for clinical trials is enormous compared to the rest of what R&D spends money on, and also I think it's highly inefficient and often gives us the wrong answer. So let me just talk about clinical measurement science. It would be a new discipline. It's a way of making sure the data are collected rigorously and consistently, and then analyzing it properly, using either machine learning, AI, data science disciplines, actually all of the above. And then these centers would provide data to key stakeholders, either to practicing physicians to make better treatment decisions in the real world, to companies trying to develop new drugs and understand how their drugs are performing in the real world, to regulators making decisions on approvability and label changes, and health economists and payers who are trying to understand the value of various treatments, particularly in the real world. Today, in clinical practice, I worry that much of the information that doctors use to make treatment decisions is gathered haphazardly and sometimes more sensitive to promotional activities rather than real data on their patients. And I also worry, but in clinical trials, what we do is we separate, for example, the examining physician from the treating physician, because we want the examining physician to make very objective measurements and provide that information for the care of patients. So at these measurement centers, you would collect data from all sources. So from radiology departments, you would get images and quantify them and store them. You would get biofluid results from various places and put them in the same place. Genetics, pathology if available, various sensor data, whether it's wearables or sensors in the home, performance tests if they're done by healthcare professionals, and then finally patient-reported outcomes. Put them all together in one place, have the data analysts there. I noticed that there's a conference in Boston where the focus area in October will be machine learning, AI, and data science computational approaches to data integration across preclinical trial and RWE. I would propose setting up these measurement centers at all major population centers, and then they would be used in clinical practice right off the bat. And then if there are new drugs to be tested, they could be basically intertwined into these measurement centers so that you would have baseline data before the patients get enrolled. See, what we do now is we have to reinvent the wheel every time we do a trial. We have to make new case report forms. You have to pay the CROs to train the physicians. We have to go and match up data with electronic health records or other medical records. It's a very cumbersome and I think costly process. And then
3 minutes is up.
Okay.
You'll get another shot. Sorry. That's it. And by the way, not everyone redoes all the case report forms. There are ways to avoid that. But we agree with that. Janet, what's your answer?
Well, I mean, I agree with almost everyone who's been speaking. I think, but I would like to roll this up. I think we have to unite as a community and insist that we stop doing clinical trials as an artisanal, one at a time, something to be well recognized in the 19th century as an experiment, and somehow drag this into the modern world where we have mass production of evidence that is rigorous and easily attained and reliable, because I think a lot of right now the results aren't all that reliable from many trials that are done for a variety of reasons. So I believe that taking all these suggestions and putting them together, you get a picture of a system. It isn't a system. It's a bunch of individual efforts. And if we want to get the evidence that our patients deserve about healthcare or about new interventions that are experimental, we've got to have a systematic approach from the ground up. And therefore, we have to raise our hands, I believe, and say, the way we do it now ain't the way going forward, and we have to change.
So I agree completely, and trying to do that. But I think that if I were to put one innovation in place, I think it's a summary of the things that everyone's talked about, but in particular, I would go with something to what Amy had talked about, and that is that we need tools and systems for evidence generation. We should not have separate systems for care and research. The practice of medicine needs to change so that we are practicing to improve, not just practicing. And if we do that and we have robust systems of data collection, most of these ills will change if we can identify early and reliable surrogates and measure them in a uniform way. We can go forward. I think this will allow us to reduce the time for drug development because all of this will count in the score that Steve is putting forward. And I think it would also be a seamless way of getting generics tested. And this, of course, all requires the new discipline of measurement science to be an integral part of everything that we do. Okay, good job, everyone. And Janet and I got our collective comments in under 3 minutes, which is
Here we go.
excellent. In 2 minutes, perfect. All right. So our second question is, what can you let go? What's your top suggestion for what we should let go, stop doing, and why? Amy.
So I think we need to let go of our emotional reaction to clinical data are of poor quality. At the end of the day, we can acknowledge the data coming out of the clinics, it has all kinds of warts. We can acknowledge that especially real-world data have challenges, but so do clinical trials data. And instead, let's move towards an approach that measures data quality in a consistent format, acknowledges that as we measure data quality, we're going to see all kinds of things that we didn't like, and then let's start to develop solutions to solve for data quality. Sometimes that is better data curation or bringing in new data sets. Sometimes that's new analytic approaches. Sometimes that is accepting the problem of poor quality data, but being able to use sensitivity analyses and other mathematical approaches to understand the overall impact. Practically speaking, though, the thing that I would give up is my emotional reaction to data quality, and what I would put in place is a structured format to start to solve for it.
Fantastic. 1 minute. I love it. Okay. Steve.
This is tough because everybody's been saying things that are music to my ears and saying it better than I might have said it. So I'll just bring in one element that hasn't been mentioned, but it's been implicit, which is in this new era of concern about reproducibility. There's a lot of clinical research that is not being properly published or reported or completely. And we know this partly through clinicaltrials.gov, which is obviously set up by our own government, but better and more complete capture of all the information that is out there, which currently we know from the empirical studies is not reaching the literature, and actually is not necessarily even reaching the regulatory bodies. We know for a fact that FDA does not see the entire clinical evidential package, and nor do other regulatory agencies. So finding better ways to follow up on some of the information we already have in clinicaltrials.gov. We know that when the studies are started, sometimes we don't know the details because they're deemed proprietary, so there's not enough information there sometimes to even track the final publication, but we can look for the clinical trials registration number. So finding the missing data, and then getting access to the data that is both negative and positive to make sure that the literature is reflecting what the actual evidence base is out there. Thank you.
Great. God, everyone's doing great. I think the timer is a good idea. Okay, Sandra, what can you let go? What do you think we should let go of that would really improve our approach to trials?
I think we should let go of putting patients through an incredible ordeal to participate in our trials. I think it starts with the consent forms that are so lengthy and poorly understood that they often don't know what they're getting into. It's the number of visits, it's traveling to the medical center, it's the number of tests, it's designs that may randomize patients to placebo or ineffective treatment. It's so many things that influence the way patients feel and function as they actually go through the trials experience. I recall a question I asked for a study that was coming forward at our portfolio committee. In this case, it was for autistic children. And I looked at the number of tests that we were putting in the clinical trial and asked, how long will that take? It would really take a full day of a family at the medical center just going through the testing. And I think that would be hard for anybody, but I can't imagine what it would be like if you were autistic. When we looked at ways in which we could advance inclusive research, particularly for people of color, we looked at prostate cancer and the out-of-pocket costs for patients to participate, the family costs, the lost time from work, all of those things, transportation, really reduce the ability to participate. One of the things I inherited now more than 6 years ago in my CMO role was a trial in inflammatory bowel disease, notoriously difficult disease to accrue patients. And we had colonoscopies for the first time that were required, as well as placebo trials in the randomization, and lots of difficulty. This trial is finally, these trials, was a series of 8 trials, have finally been fully recruited, but it took more than 7 years. So I really think we need to take a patient-centric approach to our clinical studies.
Absolutely. Okay, great job. Okay, Carl, you now have 3 minutes to say what you would like to let go of.
I harmonize strongly with Steve. We should let go of p-values. We should dismiss significance testing into the dark ages, and we should stop hypothesis testing. Statisticians have long known that the real world doesn't follow the normal distribution. Many outliers, blunders, fat tails. Significance testing is widely misunderstood. Statistical significance does not mean clinical significance or actual significance of a metric. A few years ago, more than 900 statisticians signed on to a manifesto saying, stop significance testing. I don't know if you signed on to that, Steve, but I did.
I didn't. I don't like manifestos, but I'm sympathetic.
So Steve, I think, explained elegantly the need for an evidence metric that goes beyond significance testing and the faulty lure of p-values. And I strongly support that, but I think the solution is going to be education. Steve appeared in a very amusing video a couple of years ago in a conference that he sponsored, in which a reporter went around the room and asked various eminent statisticians, what is the p-value? And almost none of them could actually explain that. You can't even explain that to a patient or to a colleague. So it's time to dismiss significance testing p-values and replace it with estimation. I favor Bayesian estimation and a consensus-derived evidence metric.
Fantastic. All right, great job. All right, Alfred, you have 3 minutes. What do you think we should let go of? What's your top choice?
I think we should reduce our reliance on performance tests that are done infrequently in clinical trials. And in a way, I'm kind of harmonizing with Sandra here. We put our patients sometime, for that day that they come in for their visit, which could be monthly or every 3 months or every 6 months, we put them through performance tests. And it is so haphazard. I mean, if the patient didn't have a good night's sleep the night before, it's effort related, there's all these factors that can introduce so much variability. And I think instead we should increase our reliance on measures that come from sensors that can be obtained from daily life. I think the nervousness about that is that we don't understand how to use these data yet. We don't understand how to separate signal from noise. But I think if we could reduce our reliance on these performance tests, it would be more patient-centric, and I think it'll improve the accuracy of our measurements.
Great. Good job. Okay, Janet, what can you let go of?
Well, I'm well known for something, and this is for all of you in the audience. We should stop doing what I call small, crappy trials. Okay, that only get you a publication, and the last sentence is always more research is needed to confirm this finding, all right? This doesn't help anyone, and yet I've talked to Francis Collins about this, and Chris Austin, who's the head of NCATS, tells me he gets pushback from the deans who say, well, our staff need these publications for the tenure committees and so forth. So we really, I think a lot of effort is spent on something that doesn't produce anything. And we really ought to sit back and think about that. I mean, it's related to this artisanal effort that we're doing now with clinical trials in general, whether the industry, but this small crappy trial, partly I think some of what Al has said, that even, and Sandra too, the industry may not always really think, and maybe the FDA reviewers want all this stuff and all this assurance, but they don't think what we're putting patients through that is crappy. And in academia, we often settle for underpowered, non-rigorous studies that really don't lead to a generalizable knowledge, and I think we should stop doing that.
I agree, because there's better ways to give credit, and again, that's part of the innovation, is like, what's the driver, right? And trying to figure out, well, if the driver is academic credit, then we should be figuring out how the deans and the chancellors promote team science. And it's not about being first author or last author. There's a very different way that we can promote this. And it's, there's always some metric of whatever is credit. Could be money, could be academic credit. And so that's a challenge that we're going to put out to the two of you to make sure that this change just speeds its way through all committees, right? Excellent. Okay. So I would say if I were going to have something let go, I would get rid of the idea of fixed randomization. I think that's really not patient-centered at all. If clearly, why are we running trials with our eyes closed, as Don Barry likes to say? If you're doing something that's an adaptive design and something is clearly outperforming the other, why should someone get something that's clearly inferior, even if it's the control? And maybe, what if the control is better? If you don't have a way to make sure that patients get something that's safe, clinicians won't want to put their patients on, and patients won't want to go on. So the idea of having a floating control, not getting rid of it, but allowing